Okay, immunology. Let's get our big show going again. Here we are. All right. So, how do these pathogens cause their pathogenic effects? How do pathogens cause disease? Honestly, there is like a full course behind every bacterium, every virus. There are mechanisms for each of these, and there are people that spend their entire lifetimes studying and trying to understand the full mechanisms of disease behind a lot of these viruses, a lot of these bacteria, and it's tricky. It's very difficult. So yeah, but these are the generalized ways, and so know that there's a lot more to the story for every single one of these than we're seeing right here. So, for example, one way that a lot of bacteria can end up damaging cells is by producing things called exotoxins. And exotoxins are little things that are released by the bacterium. Usually, they are proteins. And their job is to go out and break something down so that the bacterium can eat. So, whereas we have a digestive system that has enzymes that break things down, and we have to actually put something into the hatch at the top of our head, the mouth, in order to get it down there so it can get digested. Bacteria are kind of like inside out versions of that. So the whole world is something that they can potentially eat, and the only thing that's inside of them is their own DNA and their own proteins. So if they want to eat, their digestive surface is going to be the outside of them, and they are going to have to be able to absorb nutrients through that, and not all nutrients necessarily want to be absorbed. So you have bacteria that are on your skin right now, and they're basically feasting on you. Because to bacteria, you're a nice, warm, slightly watery pile of slowly rotting meat. All they can see is the dead part on the outside, and they eat it, and they think that's just great. Or rather, if they don't like it, hey, they can go somewhere else, right? And exotoxins. That's a big way that they're able to do this. And that's fine. It ends up cleaning off some of the dead skin cells. We keep making new skin cells, so that works out just fine. Problem is, if you get a little cut, and now some of these exotoxin producing bacteria end up inside your defenses, inside your epithelium, then they can start dissolving cells in the second, third, fourth layer, of you, and that's where they cause some really big problems. They can be very difficult to get out. Streptococcus pyogenes, Staphylococcus aureus, these are two of the things that are called flesh-eating bacteria at different times and in different places. Strep pyogenes can also be strep throat, which is essentially a flesh-eating bacteria infection in your throat. That's why you get those little red sore bits. It's because the bacteria are eating you. Yeah. Endotoxins are similar. These are things, though, that are going to have to, bacteria are going to actually burrow into your cells and then release toxins. Once again, they are trying to basically adjust the game of life to a point where they have a better chance of winning. So they're going to be breaking down your defenses. They're going to be trying to basically eat you once again, but in this case, from the inside. And then you'll have other things. A lot of viruses will work by direct cytopathic effect. So the viruses will either steal enough from the cell that the cell just kind of collapses and dies, or the viruses will sometimes actually just sort of fill the cell up and then burst out and actually rip the cell open and kill cells that way. So direct cytopathic effect is one of the ways that, for example, you can get a virus like Lassa fever virus, and if it actually gets into some of the nerves in your head, you can end up with deafness that doesn't ever go away because they've just physically destroyed the nerves that were connecting the ears to the auditory part of the brain. And yeah, there's nothing you can do about that. Polio, back in the day, used to go in, and poliomyelitis is literally destruction of some of the bodies of the nerve cells. And polio would tend to get into a little pocket and destroy all the nerve cells in that area. And nerve cells, if you know your A&P, are quite often grouped up, and you'll have a bunch of nerves going to the same place, coming out of the same branch. And so if polio were to get into that branch, it would just destroy all of those, and you would end up with paralysis of whatever is on the downstream end of those nerves. So, yeah, very much a mechanism thing that happens. Now, there are also indirect mechanisms that can lead to tissue damage. So, hepatitis B and hepatitis C viruses can do this. You can end up with immune complexes or with little bits of the virus that are floating around and are attracting certain elements of the immune system. And the immune system, these are going to be elements that would work really well at landing on a cell that's sick and destroying that cell. But when you just have free little pieces of the virus floating around, you can end up with, these complexes forming in different places and just causing kind of mayhem. They're deploying these mechanisms, but just not really attached to any one cell, and in a way that actually destroys a lot of cells. So that's bad, right? Some kinds of pathogens will have similar enough proteins or will operate in such a way, like a mycoplasma lives inside your cells, that you will end up making anti-host antibodies, antibodies against yourself. And the problem is there are a few cells that are going to be infected, but a lot of cells that are going to have your own markers on the outside. And so if, in the confusion of infection, you start to make some of these anti-self antibodies, they can then lead to, terrible conditions. Like hemolytic anemia, for example, where your red blood cells literally get destroyed. Yeah, which is not good, is it? Okay, and then, there's also cell-mediated immunity. So when your T cells actually come in and start to kill off infected cells, if those infected cells were doing something important, then you can end up having damage. So if you get a lot of infection in your liver, the only way to clear it out is by going in and physically destroying a lot of liver cells. And that's fine. It saves you from the infection, but at the cost of you not having much of a liver for a while. And so it's kind of a balance. And once again, remember that there's no overall command structure for the immune system. It's a bunch of cells individually trying to respond to danger the best way that they can with very limited cooperation. Maybe 2 or 3 cells will cooperate, and it doesn't really go much beyond that. And so, the cells don't know that, oh, you only have 10% of your liver left, and, better back off for a little bit and wait for our liver to regenerate before we go in and assassinate the last 10% that's infected. They just don't understand. So these are some of the pitfalls your immune system can fall into that would actually mean that your immune system would be doing damage to your body and not necessarily the pathogen. Kind of an indirect pathogen-prompted damage. So that's what they're talking about. Your epithelial cells provide your first barrier against infection. That just totally makes sense. You got airflow that's kind of moving things along, hopefully keeps things from having time to infect. You've got mucus that just sort of sticks everything. It's kind of like fly paper, but on the inside of your throat, lungs, what have you, nose. And you've got pulmonary surfactants that cause things to sort of slip and slide along. And the really neat ones that we're going to get to are these things called histatins and defensins and cathelicidin and reg3 proteins. There's alpha, beta, and gamma in this world. These are going to be little tiny proteins that just assemble on their own anytime there are bacteria around and punch little holes in the bacteria, because that is the general weakness of everything that's alive. Everything that's alive is a cell. And every kind of cell has an inside and an outside, and it basically stays alive by keeping the inside in and different from the outside. And if you have holes inside you, ask any shooting victim. Yeah, this is not good, right? All right. So epithelial surfaces are actually they work in a kind of a layered way. And epithelium is not just doing one thing. So on your skin on the outside, yeah, you're going to have the striatum corneum. This is going to be essentially dead cells that are just dehydrated, and they are there as a physical barrier that can be rubbed off or destroyed or whatever. It doesn't matter. We're going to make no more of them. And then inside, you've actually got a watertight lipid layer right down here at the middle. And so this is to prevent you from going into the bathtub and coming out, I don't know, 10 pounds heavier or just dissolving in a gentle rainfall. That would be bad, right? Yeah, definitely, definitely. And then you'll also have these lamellar bodies in here, and they are actually going to be one of the anti bacterial defenses that helps keep you safe. Now, down in your lungs, you're going to have the cilia. The cilia are going to have mucus on the outside, so that's this green stuff they haven't labeled. The cilia are also going to be making, rather, the cells in between the ciliated cells are going to be making surfactants to help things move along, and they're going to be making some other defenses. And then down underneath, you've got all these blood vessels. And patrolling the blood vessels are going to be white blood cells that are ready. And you've got little glands that are ready to spew out various things to help keep you healthy. Down in the intestine, I think that's really quite neat. You've got these little pockets. This is called the crypt area. And for one thing, this is where the new cells come from. So new cells are going to divide down here and move out toward the tips of the villi. But down at the bottom of the crevice, at the bottom of the crypt, you have these things called Paneth cells, and they are really good at producing things like alpha defensins and Reg3 proteins. And those are both two categories of proteins that work in a similar way. They will just land on and destroy bacteria. So in a way, you're protecting the nursery where all your little baby intestinal cells are going to be by just spewing out these little tiny proteins that will kill anything that gets too close. Yeah, like little guard dogs, if you like, for the nursery of cells in your intestines. Okay, here's some pictures. Why not? Yeah. So this is a lung epithelium. You got your nice little mucus layer up here, looking good. You got your little cilia waving along and kind of shushing that mucus, back and forth. They're doing it with a purpose. They're trying to move it in a particular direction. So we're basically want anything that lands on our mucus, which remember is like flypaper, to get shoved up and then pushed down the throat, something like that. We don't want it to stay in the lungs. That's the purpose there. Just stick it and move it out, and yeah. See you later. Go swim in the stomach acid and see how that feels. It's basically what we say to anything that lands in the lung. But unfortunately, there are some conditions like cystic fibrosis which lead to poorly working cilia. If these cilia don't function, then you get mucus there. Here's nice purple mucus all around here. But the mucus doesn't move, and so you'll get things like bacteria or fungus that come in and just then start feasting on the mucus. And you're not moving them along, and so they're just living there. And at that point, yeah, you can develop some really bad lung infections. This is one of the problems that people with cystic fibrosis face every day. And so, to start an infection, yes, those infectious agents must overcome at least one and possibly several of these innate host defenses in order to get there. So, just adhering to your epithelium, that does nothing. Your normal skin flora, which are the bacteria that you find in your skin, are there anyway. And yeah, no big deal. In your lung, on the surface, you would actually have some macrophages, some of these phagocytes, just waiting to pounce on anything that comes along. But if you get penetration of the epithelium, so if you get a little scratch, a little damage, or for like a sexually transmitted disease like a papillomavirus, just the actual friction there is going to make some little tiny abrasions, little tiny tears, which can potentially let something like a virus or bacterium get to these inner cells, which are the living ones, which are the ones that it wants to get to where it can actually replicate. Yeah, so once it's there, it can start to infect the tissues that are down here. And there are going to be cells. You're going to have tissue-resident macrophages, and you're going to have dendritic cells that are in most tissues. And both of these things, their job is to report on danger, grab little pieces of it, and go to a lymph node. go show the rest of the immune system exactly what you found. And then later on, your adaptive immunity, you can actually send CD8 T cells into this zone, and you can send new monocytes in that'll become macrophages and will try and hammer away. You can also send in antibodies, and yeah, lots of ways to prevent infection from getting out of hand. So, what is it about bacteria that makes them bad, and what are we trying to do to them? Well, a bacterium is best way I've heard it described is it's like a sausage. So you got to imagine cooking a sausage, and I'm assuming you've done this before. Remember why you poke them with a fork sometimes as they're cooking, or do you remember what happens if you don't? They split open, right? And they split open at a particular place. So generally, they are not going to split open at the curved ends because you can actually physically model this. The long, skinny sides of a sausage shaped object are going to be under the most pressure. And so, bacteria, a lot of them are sausage shaped or tic tac shaped, whatever you like. Shaped kind of like a little hot dog. And so bacteria are actually going to be wrapped around with these sort of girding layers. It's like they're wearing a girdle of cell wall, and it's really thick and tough and well joined together around the sides. And it has to be because if anything comes along and makes a little tiny gap in there, so we have lysozyme, for example, in our tears, and its job is to come in and just break apart bacterial cell walls. And if you get down to where you have an exposed lipid bilayer or even a weakened cell wall, those bacteria are actually under a lot of what is called osmotic pressure. This means that they've stored up various chemicals, ions, things like that on the inside. This is what's keeping them alive. And the concentration is different than what's on the outside of the cell. And so this creates a pressure because all of those little ions and things want to get out, basically. That's Le Chatelier's principle that you probably learned in high school physics. Basically, if you have a membrane and you have different concentrations of things on either side, those little things are going to move in such a way as to try to even out the concentration. Yeah, in this case, you've got a membrane there, and those little ions can't get past it. And so the bacteria just literally burst like a sausage. They just explode if you just weaken the outer membrane. A lot of antibiotics work this way. This is how things like the beta lactam antibiotics, they'd be things like penicillin, for example, would work this way by just breaking down a couple of these little cross links up here so that the bacteria will just explode under their own pressure. Really wonderful. Anyway, inside, so there are two different ways that bacteria are put together, or these are the two common ways anyway. One is with two membranes, so an outer membrane and an inner membrane with a cell wall made of this stuff called peptidoglycan on the inside. And it's called peptidoglycan. Glycan means sugar. Peptide means peptide, a bit of protein. And so these are little bits of protein hooked up to sugar. The proteins are the circles, the sugars are the hexagons. And yeah, it's literally linked up like that, and then they are cross-linked together. That's what these little red things are. Interesting thing out here is that on the outside, on their outer membrane, instead of it just being a membrane, they'll often have these molecules. So it's a lipid molecule with some polysaccharide on there. And so poly means a bunch of, and saccharide is sugar molecules. So polysaccharides on the outside of a lipid molecule are something that you find on what they call gram-negative bacteria. That's going to be things like E. coli, anything with those two membranes and a sort of a sandwich of peptidoglycan cell wall in the middle. It turns out that just a little tiny bit of lipopolysaccharide will absolutely send some parts of your innate immune system into just a frenzy. They are extremely pro-inflammatory. And so even tiny amounts of this, if they get into IV nutrition, for example, will absolutely kill a patient. Yeah, very, very small amounts of this. And so that's why people that are preparing these in hospitals, and if you work in one of these places or a pharmacist's office, you know about this. You know that LPS is bad, and that bad things are going to happen if there's even the tiniest trace of LPS in there. Now, for gram-positive bacteria, they're at a little more risk from the world, so they've got their cell wall right on the outside. And so some of these things, like beta lactam antibiotics, can have a little more effect on them. They can just attack them straight up. But the other thing you find in gram-positive bacteria like Staphylococcus and Streptococcus, for example, are they're going to have these things called lipoteichoic acid. And these also play a big role in not just the function of the bacteria, but in the disease that they can cause. So, how do we defend ourselves? This is where we're going with this section, and then we'll pause before the next section, which is going to be on complement. So one way we protect ourselves are with these things that are like defensins. So you can call them defensins as a group, but they're actually different categories of them. They work in a similar way. And so the way that they work is that you're going to have proteins, and the yellow and green part of the protein, they're just going to show it like an egg over here and forever in the rest of this slide set. But it's actually going to look like this. And so you got your protein here, you got a little yellow alpha helix looking great there. You've got a little 3-stranded beta sheet down here, and it's kind of a twisted beta sheet, so they're kind of rotated. And you got some, looks like random coil joining all of that up. That's what a defensin looks like. This is beta defensin, beta 1 defensin, a particular subtype of beta defensin. And the way they work is by being amphipathic. So amphipathic means they're going to have some parts that are hydrophilic, that want to be where the water is or on the outside. And they have some parts that are going to be hydrophobic. So in general, this little helix part is going to be hydrophilic, or rather it's going to have a hydrophilic side to it that's going to be the outside. That is going to tend to orient itself up here facing water. The green part down here is going to be kind of hydrophobic. And so as soon as that gets a chance, and it's going to get a chance once we cleave off a little peptide on it that was sort of keeping it in solution, but it's going to try and bury itself because it's very hydrophobic on the inside of the membrane. And so these things will naturally be attracted to a membrane. And the really neat thing is these are naturally occurring, and beta defensins are actually very primitive. So you can find beta defensins in all mammal genomes that have been sequenced, and you can even find some of these back in birds. And if they were in birds, you know, they probably go back to dinosaurs because dinosaurs are birds. So yeah, beta defensins, they go way back. Been protecting vertebrate animal life since, I don't know, 150 million BC, something like that. Anyway, once these things are in the membrane, they actually have the ability to assemble. So they will link together, and when they link together, they'll form, oftentimes it's going to be 6 of them linked together, and there's going to be a little hole in the center. And the hole is actually going to let the guts of the bacteria go out. So it's not even just that they are, going to be weakening the cell wall. They are literally going to put a hole in that bacterium if they can get to it. And they can work against other things too. They can fight fungus, they can fight different parasites, all sorts of parasites. Okay, so here are some of the major categories. You can see they're all kind of the same thing. So this thing back here, AMPH, is the amphipathic antimicrobial peptide. So the yellow bit's going to be that positive alpha helix. The green bit's going to be that hydrophobic beta sheet region. And then each of these things is going to have a little pro region up here. So it's a little part of the protein that's going to fold up, and it's actually going to kind of bind around this hydrophobic bit and keep the whole thing in solution. Because if it's too hydrophobic, you're not going to be able to make it outside of a cell. It's just going to live in the cell's membrane. And of course, if they all click together in a cell's membrane, then that cell is going to be dead. So we need a good defense against this. This is something we're trying to use against invaders, and we don't want it to be used against ourselves. It's like keeping a gun in the home, right? Yeah, same sort of thing. So this is the safety mechanism, or the gun lock, or the gun safe, whatever, if you want to go with that particular metaphor. And so there are alpha and beta defensins. There are cathelicidins, and which have a cathelin molecule up here at the end, but are still cut and activated, and the amphipathic region is the one that will go out and, do some damage. And then there are the reg3 proteins, reg3 alpha, beta, and gamma, and there are subtypes of most of these things that are produced in different cells. So we have lots of ways to make slight variants of this thing that'll work a little bit better or a little bit worse against different kinds of potential pathogens. And so, yeah, by just keeping up this constant barrage of things that will kill bacteria, we can help to at least limit the spread of bacteria and keep them out of places where we do not want them, basically. Other interesting thing here, you see these little black connectors on the top. Those are what are called disulfide bonds. So the amino acid cysteine has a sulfur group at the end of it. It's actually an SH molecule when it's reduced. And if you get two of those in the same molecule, they can link together and form a sulfur to sulfur covalent bond, which is what we call a disulfide bond, because there's two of them. Yeah, makes sense, right? And so they will have three of them in here. And it's actually been shown that these amphipathic proteins work better when those disulfide bonds are reduced. So generally, the area outside of a cell is going to be kind of oxidizing, and so these are going to be linked up as disulfides outside the cell. But things like alpha and beta defensins are going to work really well if a pathogen happens to be making a reducing atmosphere around itself, a reducing sort of environment, or they'll work really well if they get inside of a cell. So these can actually track down some of those bacteria that try to burrow into our cells, or things like malaria parasites, potentially. They could go after those. And here is the ultimate goal shown down here at the bottom. These are some of the little hexagons, and the hexagon is made of 6 copies of one of these defensin type proteins. And so there you got your little helix up there, and you got your big beta sheet at the back. This is a different one than the one we were looking at before. This one has 2 helices. But this is how they work. They click together inside the membrane. These have been purified, so you can just see them floating around. But in the center there, you've got a hole, and that hole is for real. And that is where the guts of the bacterium will leak out. So yeah, we don't shoot bacteria, but we do fill them full of holes in actually several ways, this being just one of them. So there we go, and that's the end of that section, and we will go on and talk about complement in the next section. Thank you very much.